In this video, we will discuss G-protein coupled receptors and a couple examples of signaling pathways associated with G-protein coupled receptors. The learning objectives for this video are to become familiar with the different components of G-protein coupled receptor signaling pathways, understand the different signaling pathways initiated by different G-proteins, and appreciate how different G-protein coupled receptor signaling pathways alter physiology. There are several different G-protein coupled receptors, or GPCRs, which can activate several types of G-proteins, which in turn have different signaling pathways. This diversity is key as G-protein coupled receptors play a major role in many physiological functions of the body, from interpreting messages in the forms of ligands, from peptides, sugars, light, and more. These signals reflect the environment and nutritional state a eukaryote is in and helps with survival and homeostasis. Because of this wide variety of targets, over 1/3 of drugs on the market target and modulate GPCRs. G-protein coupled receptors are 7 transmembrane proteins. Once a ligand binds, the receptor goes through a conformational change and activates a guanine nucleotide binding protein, also called G-protein. To activate the G-protein, the bound GDP is exchanged for GTP. After activation, the G-protein and active subunits are released to trigger their cellular action and will remain active until the GTP is hydrolyzed to the inactive GDP, once again deactivating the G-protein. This is important to note, as the release of the ligand may not necessarily mean that the G-protein is deactivated, because as long as GTP is bound, the signal will continue. Here is an overall schematic of GPCR signaling. As you can see, there are many types of ligands GPCRs can bind. Once bound, the G-protein is activated. G-proteins are made of alpha, beta, and gamma subunits. The alpha subunit is the subunit bound to GDP until the receptor triggers the binding of GTP. Once GTP binds, the alpha subunit starts a second messenger system, while the beta-gamma subunits activate other effector proteins. The second messenger system activated correlates with the type of G-protein alpha subunit activated. For example, the first alpha subunit listed here is G-alpha-S, which stands for stimulatory since it stimulates the activity of adenyl cyclase, which in turn increases levels of intracellular cyclic AMP. The overall effect of the effector protein is to change the intracellular contents to signal to the nucleus to alter gene expression, and to start a biological response. In the next 2 slides, we will go into more detail about G-alpha-S, G-alpha-I, and G-alpha-Q. As mentioned previously, the S in G-alpha-S stands for stimulatory, while I stands for inhibitory. Key components for this signaling pathway are the appropriate ligand for a given GPCR, G-alpha-S protein, or G-alpha-I protein, adenyl cyclase as an effector protein, and either the increase or decrease production of cyclic AMP. Cyclic AMP will activate pKa, which is the protein kinase A, and will in turn phosphorylate many enzymes and proteins, resulting in an overall effect on gene transcription and protein activation. G-alpha-S and G-alpha-I often have opposing effects within the same physiological system. For the G-alpha-Q signaling pathway, phospholipase C is activated by G-alpha-Q. PLC converts the phospholipid PIP2 into DAG and IP3. Though the simplified diagram does not show it, DAG stays membrane bound. DAG activates PKC, a kinase that phosphorylates many proteins. IP3 is freely soluble in the cell. Once it reaches the endoplasmic reticulum, or sarcoplasmic reticulum in muscle cells, it binds ligand-gated calcium channels and releases a large amount of calcium into the cytoplasm. High levels of calcium can trigger many signaling pathways, including pathways regulated by calcineurin. The G-protein beta and gamma subunits also play a role in GQ signaling, activating pathways that will complement the G-protein alpha pathways. There are several G-proteins, including GS, GI, GULF, GO, GQ, and GT. In this discussion, we covered GS, GI, and GQ. Some specific ligands and G-protein coupled receptors are listed here, as is a summary of the effector and signaling pathways we talked about. As you can see, some adrenergic receptors are activators of GS, while others activate GI, Creating a negative feedback loop, we will cover in more detail in the next few slides. Let's think about an example of an agonist and an antagonist commonly used in veterinary medicine, called xylazine and yohimbine. First, let's look at what naturally happens in the body. Here is a diagram of a synapse between two neurons. The presynaptic neuron has an alpha-2 G protein coupled receptor on its membrane, while the postsynaptic neuron has beta-2 G protein coupled receptors. When a signal is initiated in the sympathetic nervous system, potentially from getting scared, norepinephrine is released and binds to beta-2 adrenergic receptors, which continues the signal onward, resulting, for example, in an increased heart rate. Some of the released norepinephrine will act as a negative feedback loop by binding the alpha-2 receptors, which results in a decrease in cyclic AMP, which prevents further release of norepinephrine, halting the signal. Now let's look at what happens when you give a dog xylazine. Xylazine acts like norepinephrine and is therefore an agonist for the alpha-2 receptor. Xylazine binds, resulting in a decreased concentration of cyclic AMP in the presynaptic neuron, regardless of signals from the presynaptic neuron, which in turn prevents the release of norepinephrine, resulting in no signal reaching the postsynaptic neuron, and your dog becomes sedate. But now you want your dog to wake up from sedation. You want him up and walking out of the vet's office. To reverse the effects of xylazine, you can use yohimbine. Can you guess how yohimbine counteracts the signal caused by xylazine? Yohimbine is an antagonist for the alpha-2 receptor. Therefore, yohimbine blocks the receptor activity, resulting in an increase of cyclic AMP in the presynaptic neuron, which triggers the release of stores of norepinephrine, which then can act on the postsynaptic neuron to trigger a response to wake up your dog. This example has been simplified, but the take-home message is that agonists and antagonists can act on the same receptor to modify the release of endogenous ligands, such as hormones, to cause a whole body effect. It is interesting to note that the agonist causes a decrease effect while the antagonist causes an increase effect in this example. This is because the alpha-2 receptor inhibits a signaling pathway. Make sure to understand which receptors are activating and which are inhibitory.